Do we need to?
So let's take a look at now anti-cancer drugs. So
Okay.
each stage, G1, S, G2. So we're going to start at G1.
I wanted to ask you where you wanted to contribute to this.
Um, G1 and the final phase G2. So where are we starting? G1.
I wasn't going to do it in this way.
Okay.
I think, so we're going to explore some of the anti-cancer drugs in this next section. There are three approaches for when you're trying to treat cancer. You have your surgical approach of direct extraction. You have radiological approach using radiation to target the cancer for destruction, and you have your chemotherapy approach using medications, some of these cytotoxic drugs to try and selectively kill the cancer cells. When we're looking at cytotoxic drugs, we want a drug that can have selectivity for the cancer cells over the normal human cells. And that's what happens here and that's what's important here because the cancer drugs and the human drugs, the cancer cells and the human cells share a lot of common factors because the cancer cells are just are literally human cells that have undergone a lot of mutations and a lot of these cancer mechanisms that have allowed them to become, because as cancers develop, they become more de-differentiated, more stem cell like. So, but they are from the same origin, so a lot of them will express the same things. So it's very hard to selectively target the cancer cells. So when we're looking at chemotherapy drugs, we're looking at drugs that can get directly into the cell and have cytotoxic effects. So a lot of these will have interactions with the DNA. Now, we have the hallmarks of cancer by Hardy and Weinberg in 2000. These allow us to have a little bit of a targeting of cancer cells rather than normal cells. So the thing I will be touching on quite a lot in this is something called, it's not the exact term in this case, but use dependence. Use dependence is where you can target with a drug something where there is different levels of activity. So a high use versus a low use. So things like local anesthetics, they target they target cells with high firing, high frequency of firing. So in cancer, you have a very high proliferative rate. You have a very high proliferative rate, and that's the crucial determining factor where you can target cancer cells with some of these treatments. So I think it's very important to touch upon the cell cycle because what we're trying to do with a lot of these cytotoxic agents is induce DNA damages. That's one of the primary mechanisms of how we're trying to target these cells. The other ways that we can do that is also targeting things like DNA. We can either cause direct DNA damage, target the DNA integrity, or try and target elements of mitosis itself, such as the spindle. So in this way, you're trying to set up damages to the cancer cells, and you're hoping that these get picked up by the cell cycle machinery and these different checkpoints. There are different checkpoints throughout the cell cycle. You have G0 to G1, and when you switch between the different stages of G1S, G2, you have checkpoints to assess and to make sure that the DNA replication, everything is undergone correctly so that you can ensure genomic stability. So yes, so you're priming, you're causing these damages to be picked up by the machinery. Now this can sometimes go wrong because of the fact that cancer cells, one some of their hallmarks is the fact they can downregulate apoptotic pathways so they can survive. That's one of the things. The other thing is you keep giving these cancer cells mutations and damages, and sometimes they just don't care. And so they can just bypass some of these checkpoints. So that's where the targeting comes in. It makes it quite hard. But let's break down how the cell cycle really is beginning. So there's just the general concepts. You have growth factors. These factors are activating different receptors on the cell surface. These will most likely be tyrosine kinase receptors. So an example of a pathway here is epidermal growth factor, binding to the epidermal growth factor receptor. The tyrosine kinase transphosphorylates itself and dimerizes, and then has downstream signaling with RAS, different kinases, MAP kinase, all the way to have cytosolic transduction, and then to go to the nucleus, have nuclear transduction, and then transcribe different genes. So a lot of these will be cell cycle proteins, things like cyclins, CDKs that are
Mm.
involved for cycling between the different stages of the cell cycle. And in these pathways, you have two different types of proteins or mediators. You have oncogenes and you have tumor suppressor genes. Have you heard of these?
Briefly, yeah.
So your oncogenes are ones that are driving the cell cycle. Your tumor suppressor genes are the ones that are kind of slowing down the cell cycle, inhibiting a lot of these agents.
Yeah,
So it's a
translations.
balance of activation and inhibition of the cell cycle is where you get this optimum and you have the normal cell growth. And if you have mutations in these genes, which is what happens in a lot of these cancers, different stages of these pathways, you have your oncogenes having gain of function mutations where they become hyperactive and you keep pushing through the cell cycle. And you have tumor suppressor genes where if you lose functioning, this is more often a recessive. So you have two copies of each of these, and oncogenes are uh, you need one copy to start activating and driving the cell cycle. Tumor suppression, if you both copies, you have none of this safety mechanisms, none of this inhibition. So you just can't downregulate the cell cycle, and that's how you get high proliferation. Okay, so I think let's get stuck in with some of the nitty gritty of some of these drugs.
Alright.
So the first thing we have is impairing the DNA replication. And here you're trying to arrest the cell cycle at a lot of these checkpoints. So they pick up damages and things like that. You start activating your different pathways, picked up by things like P15, P16, P21, and then having P53, which is the guardian of the genome. This one gene, P53, will encode a protein and it shuts down and it starts apoptosis. So that's a key mechanism there. So first we have a lot of different agents. I'll mention start talking about cross-linking and alkylating agents first. We have nitrogen mustards as our first step. These are alkylating agents. So these have. Do you want to get a picture of some of these? If you want to throw up a diagram of how these look like. Uh, but you have these chloroethyl groups, two chloroethyl groups, and what will happen is you'll these very reactive electrophilic groups. So the first thing that happens is the first chloroethyl groups will react, and you'll have a cyclization of the nitrogen mustard first. So, so you have the cyclization, which will form your ammonium ion. This ammonium ion, again, you have a spare chloride, a negative chloride um, group, which is very electrophilic. So it'll attack the O6, the oxygen position 6, or the N7 of guanine, and therefore and then you have another group, chloroethyl group, because there are two chloroethyl groups. So you can attack two different guanines, and this is what causes your inter-strand cross-linking, but also intra-strand cross-linking. So you have cross-linking on one strand of the DNA and between the two strands. And what this means is
Mhm.
that you're distorting the structure and the architecture of the DNA. The major and the minor grooves, which are usually there, that help things like DNA polymerase bind so you can start replicating your genome, can no longer do that. The other thing that can happen is because you have two chloroethyl groups, if one chloroethyl group reacts with water, then you can have a GC to AT transversion. You have mispairing, and when the cell cycle machinery tries to fix it, then you start getting then you have this transversion from faulty machinery. Trying to say something?
Yeah, I was just gonna ask about the chlorine. You said that chlorine was an agent like electrophilic attacks, right?
Yeah.
So the chlorine specifically, and this is the case in many other areas of the body, is triggers a lot of these like bad reactions in the body that can lead to
Yeah.
cancer directly. Cytotoxicity in particular. Okay.
Yeah. So yeah, chlorine, very electrophilic group, highly reactive. So if you start having nucleophilic groups around, which are very, very abundant, things in DNA, RNA, even proteins. That's what makes a lot of these nitrogen musters highly toxic because they will start reacting with these other different proteins, other nucleic acids, and a lot of off-target effects, very non-selective in this way. I think it's also quite there's different types of the nitrogen musters. You have cyclophosphamide, which is a prodrug. It needs to be involved in liver metabolism. An important thing is you have hemorrhagic cystitis because because breaking down cyclophosphamide produces something called acrolein, which causes a lot of bleeding from can
Very
be
dangerous.
deposited in the bladder and cause a lot of bleeding. So yeah, the other thing that I think is really cool is you can try with some of these cytotoxic agents because they're very non-specific, make them a bit more specific. You have something called melphalan, where you can target melanomas, so skin cancer, because these will have a lot of pigment melatonin, and to make melatonin you need phenylalanine. So if you have add a phenylalanine group onto this nitrogen mustard, then it can selectively go to that type of cancer, that cell type. Okay, next we're going to touch upon the nitrous Again, similar concept. They are alkylating, but they're also carbamylating agents. So they have alkyl groups and carbonyl groups to interact and cause your cross-linking. Now these are more targeted towards the O6 rather than the N7 of the guanine. Similar mechanism though, similar process. There's different types. You have carmustine and lomustine, your steam drugs really. These are more lipophilic. They have, I think, it's a quinone group. No, they have chlorethyl side chains, very lipophilic. You can use them in the brain for brain cancer, more specifically. And you also have streptozocin. So the similar way how I said you can get phenylalanine, you can have phenylalanine group on a nitrogen mustard to target melanomas. If you have pancreatic islet cancer, so what cells would I be talking about here? Which cells secrete? Which cells are monitoring your glucose levels?
Beta cells in the
Yes,
pancreas.
so they'll be secreting your insulin. These will have GLUT2 receptors to get glucose into these cells, and so streptozocin, streptozocin, uh, can is a glucose analog, very similar structure to glucose. You can get it into those cells more specifically. Okay, then we have other agents. We have mitomycin C and cisplatin. Mitomycin C is an alkylating agent, similar again, and it inhibits DNA synthesis. And the what they found with mitomycin C was it actually needs to be metabolized and needs some enzymatic reduction within the cell, within the cancer cell, to turn it into its active form. Cisplatin, the real breakthrough drug for testicular cancer, actually, which I want to talk on this, actually really interesting. Trying to use old drugs with new tricks. So yeah, so they were trying to use cisplatin in different contexts. But yeah, so cisplatin has
Toxic effects.
I've written a lot of jargon here that is actually not helping me out at all. Um, Cisplatin, because of its really quite planar structure, it's mainly in your interest around cross-linking. It's very involved in this geometry of the DNA, and so having these interests around constant causes these bending, and it leads to a concept called steric hindrance, where physically the shape of the DNA doesn't allow things like DNA polymerase, transcription factors to bind, distorts the major and the minor groups. So in this way, cisplatin causes a lot of these DNA damages and does not allow for replication. Now, the other thing that we have is also intercalating agents into the DNA. So the two main types of drugs here are your anthracyclines, so your doxorubicin, daunorubicin. You also have actinomycin D, and these have benzene rings. So they have very planar structure with very so the benzene ring itself is has these pi uh these
Alternating sigma and pi orbitals.
yeah, these
Alternating double bonds.
above and below the plane of the ring. So it's quite reactive, quite easily get in and react with things. So what you have in these very flat molecules that start intercalating in between the base pairs of the DNA. And as you can assume, a lot of this is involved in uh, altering the shape of the DNA, changing the sequence. So yeah, in that way, causing DNA damage. What they also found was that in particular, was uh, the fact that you can also have a lot of interaction with things like DNA topoisomerases. So then they affect the unwinding of the DNA, so they have those effects. They also involved in having a lot of reactive oxygen species and a lot of damage to the DNA in that way. However, this does have a side effect profile, particularly with cardiotoxicity. So that's why they developed a metoxantrone, which doesn't produce these reactive oxygen species, so it's less cardiotoxic. So it's a better side effect profile. Okay. Again, I mentioned another drug. There's the last of this section of the intercalating agents. You have bleomycin, which is an iron chelating agent, metal chelating agent. Iron 2 plus or copper plus, these reduced metals, they form into the structure of this glycopeptide, and they cause DNA breaks, the DNA fragmentation, because they interact with the ribophosphodiester backbone. So they break it up into really smaller chunks. And they do this by forming superoxide radicals. These reduced ions interact with oxygen, forming these superoxide radicals. Now, the last two areas we're going to touch upon with these anti-cancer drugs and these chemotherapeutic agents are we're going to look at metabolites and antagonists of the building blocks of the DNA, and then we're going to look at the cell cycle assembly and looking at the spindle type of drugs.